Okay, shall we start then the session? Yeah. Um, any questions you have regarding last two sessions? Some questions were asked. I will be trying to answer those questions today at the end of this session. And we will be talking today about quantum dots and carbon dots mainly. And for quantum dots and carbon dots, they are fluorescent molecules. So for their characterization, you need a very important spectroscopy technique, which is known as photoluminescence spectroscopy technique, which I don't think has been mentioned in biomedical nanotech course. Do any of you know PL spectroscopy? Basics of PL spectroscopy? Okay. So we will be doing a short recap of PL spectroscopy, basics of PL. for what is photoluminescence and how actually the you get the fluorescence in carbon dots and quantum dots. Little bit we'll be talking about semiconductors, because quantum dot is a semiconductor material. So it will so we will have certain terms like valence band, conduction band, and a little bit of physics terms. So in case you do not understand, please ask questions. Please ask me what you did not understand, and uh, I'll try to help you understand as much as possible. So shall we start the session then? Okay. Can you see the presentation?
Yes, ma'am.
Okay, thank you. So today we will be talking about, like I said, we will be talking about quantum dots. So I guess you have already been given an overview what quantum dots are. These are basically semiconductor nanocrystals. Can anybody tell me what is the size of quantum dots? Size of quantum
It
dots.
varies. It varies from 3 to 10 nanometers.
Yes,
Uh,
three, or you can even say one or two nanometers. Yeah. So, difference between quantum dots and carbon dots. Difference between quantum dots and carbon dots.
Carbon dots are their shell is completely made of carbon only, and quantum dots
Yes.
can vary from inorganic to organic based on the core element used.
Yeah, core elements. So, what kind of elements are mostly used as core elements?
Transitional elements.
Yes, transition elements, correct. And these transition elements, they are mostly heavy metals, so they're toxic in nature. So I think I mentioned in the previous session that the reason why like carbon dots are most nowadays used mostly compared to quantum dots. Even quantum dots are used. I will give you a very yes. I will give you a very uh, like regular example. You might have noticed in everyday life, some people might even have it in your home. So that is an example of quantum dot technology. So so let's talk about quantum dots first. You can see the presentation again. Can
Yes,
you?
ma'am.
Okay, okay, thank you. So, quantum dots are semiconductor nanocrystal. So, what do you understand by semiconductor? We have three kinds of materials, right? One is an insulator, which does not conduct any kind of electrical charge. One is a metallic metals, and another we have semiconductor. So, metals, they have free electrons. That is why they conduct electricity. And semiconductors, do they have free electrons?
They have holes and electrons both.
No, 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 no. They do not have holes and electrons. Holes are generated in semiconductors. So
Yeah.
basically, yeah. So basically, what happens in semiconductor material is metal is you have the valence band and you have the conduction band, so they're very close to each other. Okay, so your valence band electrons, they can freely move around in the conduction band electron, so they have a lot of space to move around. So whenever you apply an electric charge, it will pass electric current very easily. Whereas for semiconductors, what you have the valence band and you have the conduction band, and there's a gap. That gap is called band gap. Okay, so basically you need, depending on the band gap value, you need that much amount of energy to excite an electron from the valence band to the conduction band. Okay, so when you excite an electron from the valence band to the conduction band, then the electron is excited from valence band to conduction band. So in place of the electron in the valence band, there is there is an empty space that is known as a hole, which carries a positive charge. That which is basically a hole is basically absence of negative charge. Okay, so that is how semiconductors, they conduct electricity. So if your band gap is too high, then you need more energy to excite electrons to conduction band. As a result, it becomes more difficult. Like conductivity of that semiconductor is less compared to semiconductors which have smaller band gaps. Okay, everybody understood this concept of difference between metallic and semiconductors?
Yes, ma'am.
Okay, so basically what you have is you have uh, you have in this quantum dots, these are semiconductor nanocrystals. So basically you have a conduction band and a valence band. One second. Okay, so basically you have a conduction band and a valence band, and you have to excite electrons to the conduction band in order to see fluorescence. So this quantum dot is basically composed of elements from group 2 to 6, such as some examples are cadmium selenium, cadmium sulfate, or cadmium tellurium, and group 3 to 5 such as INP or INAS, or groups 4 to 6 such as lead selenium. So as you can see, these are all heavy metals. So the quantum dot core, as you can see here, is made of very like toxic heavy metals. The shell is usually composed of ZnS. So you have a shell which is made of ZnS. So why do we have this shell? Why do we have this shell? Any idea why we have the shell?
For the protection?
No, it's not for the protection. The shell is to improve your fluorescence property. So it will increase the quantum yield. Basically, it will give you more fluorescence. That is why they're adding the shell. So see, it's written here, the shell is usually composed of ZnS to improve the optical properties. So the fluorescence yield will be more if you have a shell. Okay, so that is the reason you have the shell. Then you have this, can you see this hydrophobic surface ligand? These black dots with tails. Can you see these hydrophobic surface ligands, black dots with tails? These are basically caps. So now the thing that you were saying that the shell is for stabilizing, these caps are basically for stabilizing. So this is this will stabilize it, as well as sometimes for biology related work it will, because its quantum dot is basically, you can see these are transition metals. So to make it more aqueous soluble, as well as to make it more biocompatible, you have this cap. With this cap you can do further modifications depending on what kind of capping agent you're using. You can have different kinds of drugs loaded onto, you can attach the drugs to these caps. You can put hydrophobic drugs, as you can see here, you can load hydrophobic drugs. You can attach proteins to these quantum dots for target for some kind of transport. You have these are amphiphilic polymers attached. The like this is functionalization basically. So quantum dot can be further functionalized. You can attach antibodies for targeted delivery of some drug. So like this, you can further functionalize the quantum dot for different applications. Okay, so basically, did you understand the structure of quantum dot? How what you will need for quantum dot? You need a quantum dot core, you need a shell, you need a capping agent, and then further functionalization depending on your application. Is it clear? So next we will be talking about that how are quantum dots, they're different from bulk semiconductors. So here is an example you can see. This is your bulk, this in black represents your bulk semiconductor, and you can see the valence band here and the conduction band here on top. So VB is valence band, CB is conduction band, and you can see this distance between the valence band and the conduction band, what I said, this is called band gap. So this band gap you can see is low in bulk semiconductor. But when you are going to quantum, dots, when you're going to nano range, as you can see, with decreasing quantum dot size, the band gap is increasing. So here represented are from 6 nanometer. This is 6 nanometer. This is the red one. The yellow one is 4 nanometer, and the green one is 2 nanometer. So with decrease in size, you can see that the band gap is increasing. So this is due to a phenomena which is called quantum confinement. So basically, what is quantum confinement? Here, the electrons in your quantum dot, the electrons are confined. That is, they occupy less space compared to the bulk. Hence, the valence band and the conduction band potentials, they are shifted more positive and negative respectively. As a result, with decrease in size, the more smaller the size, the more the electrons will be confined, and hence the more your valence band and conduction band potentials will be shifted to positive and negative, and the band gap will increase. Did you understand? So, to understand quantum dots, you need to know about a term called exciton Bohr radius. So, what is this exciton Bohr radius? Like I was saying, in semiconductor materials, this is your this lower one is your valence band, the upper one is your conduction band, and this is the band gap. So basically, you need this energy, this gap energy, band gap energy, to excite an electron to your conduction band. So when you're exciting an electron to your conduction band, there will be an absence, the absence of electron in the valence band. This is a hole, and this excited electron and hole forms a pair. This distance, average distance between an electron and a hole, this is called the exciton Bohr radius, also referred to as de Broglie wavelength of the electron. Okay, so this exciton Bohr radius is very important because it plays a role in the quantum confinement phenomena. So what is happening when we are reducing the size? So in a semiconductor crystallite whose size is smaller than twice the size of its exciton Bohr radius, okay, the excitons are squeezed, leading to quantum confinement. So you understand now what is exciton Bohr radius? Any queries regarding the exciton Bohr radius? What is the exciton Bohr radius?
No, ma'am.
Okay. So basically this exciton Bohr radius, when your semiconductor crystallized, basically when your quantum dot size is like smaller than twice the size of its exciton Bohr radius, then the excitons, which are the excitons? This pair, this electron hole pair is called the exciton. So this excitons, then they're squeezed in a small space. This leads to quantum confinement. So a particle behaves as if it were free when the confining dimension is large compared to the wavelength of the particle. So when you have a bulk material, then it can behave freely. But when you have quantum dot, which size is very small, then it will become confined. So one second. So basically, during this state, the band gap remains at its original energy due to a continuous energy state. So this is the condition. This is the condition in the, you can say, in the bulk material. So when your dimension is very large compared to the wavelength of the particle, then the band gap will remain at its original energy due to a continuous energy state. However, as the confining dimension decreases and reaches a certain limit, typically nanoscale, the energy spectrum becomes discrete. So as you can see in this picture, that here you can see the energy levels, they are becoming discrete. As you're going more towards the nano range, more smaller size, you're getting more discrete energy levels. So the energy spectrum becomes discrete. As a result, the band gap becomes size dependent. This ultimately results in a blue shift in light emission as the size of the particle decreases. Now you can understand why with decreasing size you are slowly getting blue emission. First red, then yellow, then green, VIBGR basically. So you will ultimately get the smallest particle size will give you blue emission. So this is known as blue shift. So smaller the size, the more your spectrum will shift towards the blue range of the spectrum, and your band gap will increase due to a phenomena called quantum confinement. So let's read a little bit about quantum confinement, what it is. So it says the smaller you make the crystal, the higher the energy of the electron will be. We already know that when you go to the nanoscale level, they are very very reactive. So similarly for quantum dots also it is the same case. The smaller crystal you make, the higher energy the electrons will have. So the kinetic energy of that electron will get increased. Now we know uncertainty principle. It states that the more precisely one knows the position of a particle, the more uncertainty in its momentum and vice versa. So basically you cannot, uncertainty principle says that you cannot know the position as well as the momentum of the particle simultaneously. Either you know the position of the particle or you know the momentum. So therefore, the more spatially confined and localized a particle becomes, the broader the range of its momentum or energy. This phenomenon of increasing excitation energy with decreasing crystallite size, this is known as quantum confinement. Okay, this is more in scientific terms. This thing, it was more in simple terms, basically what happens, why the band gap increases. So this is more in scientific terms. So did you understand it? What is quantum confinement? What is happening? How the band gap is increasing? How your with size you're getting a blue shift in your quantum dots? Any queries? Any questions? Okay, okay. So basically, this what do you mean by increasing excitation energy? Previously we were talking about band gap, right? Like with decreasing crystallite size, basically decreasing quantum dot size, there was an increase in band gap. So what do you mean by this increasing excitation energy? What do you mean by this excitation energy? Any idea?
The energy provided for exciting the electrons, that it jumps from the valence band to the conduction band.
Yes,
So minimum
that will
energy
be,
required.
yeah, minimum energy required, that will be equal to the
EV.
EV or EG, band gap, EG.
Ha,
EG.
EG.
Yeah, band gap. Okay, thank you. Uh, so basically yes, so this is what is quantum confinement, that with decreasing crystallite size, you are increasing excitation energy or increasing the band gap. Okay, this is known as quantum confinement. So this is an example of application of quantum dots in sensing. So as you can see here, you have two different colored quantum dots. One is green fluorescence, one is red fluorescence. And you have basically taken a cell which has a tumor marker. So this tumor marker, you are targeting first a primary antibody to this tumor marker, and then you are attaching your quantum dots to your secondary antibody. Okay, so the primary antibody, when it goes and binds to the tumor marker, the secondary antibodies which is tagged with the quantum dots, they will go and bind to your primary antibody, and it will on excitation, like these quantum dots, they will have a particular excitation wavelength. When you excite them, it will give fluorescence at a particular at a different wavelength. So and as a result you will be able to visualize the cell. So if this will be very specific, because if this antibody binds specifically only to this tumor marker, then only the cells bearing this tumor marker will be detected. Whereas if cells they do not have this tumor marker, then the primary antibody cannot bind. As a result, the secondary antibody tagged with the quantum dots will also not bind, and your normal cells which do not have this tumor marker will not be detected. So it will be very specific. It will be very specific for only it will be very specific for only tumor cells. So it will specifically help in sensing or imaging tumor cells. So next one, there is another example for quantum dot. Here what they are doing is they have taken a quantum dot. This surface is basically, this is they functionalized the quantum dot with some functional groups, so that they can attach this miRNA binding sequence basically to this quantum dot. So what is happening in this miRNA binding sequence, you can see. So remember in last class I was talking, I gave an example where you were conjugating to CNT. There was CNT which was coated with polydopamine. Yes, carboxylic acid surface modification. So, what kind of reaction will you use for conjugating with the carboxylic acid and this? Any idea? No, 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 not quenching. Quenching is a phenomena. Conjugation chemistry. What kind of conjugation chemistry you will be using? I discussed in last class, I was giving that example that I gave an application of carbon nanotube sensing, in which you had the carbon nanotube, you polydopamine coated it, and then you conjugated folic acid onto the polydopamine. What kind of chemistry was used? Do any of you who were there in the last class, do you remember? No, not coordination chemistry. It is called EDC NHS chemistry. So using that EDC NHS chemistry, it's a conjugation chemistry basically. You will activate these carboxyl acid groups and this amine groups, and then they will form a amide bond, CONH2. And this miRNA binding sequence will get attached to your quantum dot surface. Do you understand now? Is it clear? Okay. So, sorry. Basically, you have this, and you can see that apart from this miRNA binding sequence, so you can see this area where it is written miRNA is where your miRNA will come and bind. Okay, and this quencher is basically there to quench your quantum dot fluorescence. Okay, so whenever you're illuminating whenever you're exciting this quantum dot, the emission will be absorbed by this quencher, and you will not get any kind of fluorescence when your miRNA is not present. But when your miRNA is present, oh, also you have this R9 peptide. So this R9 peptide also you can see has been attached to the surface of your quantum dot. So this R9 peptide basically helps in cell, like to enter the cell. This R9 peptide helps the quantum dots to enter cells. So this, when your miRNA target is present, it will come and bind to this area. It's miRNA binding sequence. As a result, it will displace this quencher. So as soon as your quencher is displaced, remember I also talked about the fret phenomena yesterday. So basically, when your quantum dot, when your quencher was near to your quantum dot, when you are exciting your quantum dot, the emission was absorbed by the quencher via the fret phenomena, same. But when your miRNA is binding and displacing your quencher, then the quencher shifts far away from the quantum dot. As a result, the fluorescence is turned on. So this is basically an on and off method of detecting your target miRNA. Very simple, on and off only. You do not have to measure intensity and all. Just on and off. On means your miRNA target is present. Off means it's not present. And this FRET, like if you remember last day I said that FRET will only work when your quencher and your fluorophore is very close to each other at 10 nanometer or less, at a distance of 10 nanometer or less. So here you can see the distance has increased. As a result, FRET phenomena is not happening and the quencher is not able to absorb the emission wavelength of the quantum dot. As a result, the quantum dot is switching on. Is it clear? Okay. So next we will be talking about carbon dots. 1 second. Next, we will be talking about carbon dots. So basically, carbon dots are also like quantum dots, same, less than 10 nanometer in size, and they're small carbon nanoparticles. You can form carbon with some form of surface passivation. Do you know what is surface passivation? Do you know what is surface passivation? Okay. So basically, surface passivation is this is a technique done so that your carbon dot, it will, if you do not have, if you do not perform surface passivation of your carbon dots, basically you attach certain groups on your carbon dot surface so that they do not lose their fluorescence properties. Like there's a photo bleaching which happens, it will slowly lose its fluorescence properties. In order to save your carbon dots from losing the fluorescence properties, this surface passivation is done. Basically it just attaches some functional groups onto your carbon dot surface to make it more stable. Is it clear? Any queries so far?
And what's the difference between surface modification and surface passivation?
Okay. Surface modification. 1 second. Okay, yeah. So surface modification is basically surface passivation is something you're doing while you're synthesizing your material. Surface modification, after your material is synthesized, then you are modifying the surface. Okay, so surface passivation is basically like when you are synthesizing normal metal nanoparticles, you are basically adding a stabilizer to stabilize your nanoparticles so that they do not aggregate. So surface passivation is also kind of like that you are adding some compound. Sometimes if you're synthesizing carbon dots from natural sources, they you have taken some plant extract and you are synthesizing carbon dots from plant extract, they will have some natural stabilizers. So that time you do not need to do any kind of surface passivation. But if you are chemically synthesizing, you are using a chemical, that time you add another chemical that will act as a stabilizing molecule for your, which will act as a surface passivation for your carbon dot. So that is surface passivation. And surface modification is after you have synthesized your material, what kind of what is your application of your material. So depending on your application, you will make modifications. Is it clear? Okay. Sometimes what happens is that you can also use the same molecule which you want, like you want to, suppose you want to conjugate something. So as a surface passivation, you can already, like you want to conjugate antibodies to your carbon dot. So you can already add something which has a carboxyl group so that your carbon dot will represent the carboxyl group on its surface. Okay, so that's like, and then you will do the EDC NHS chemistry to attach your antibody. So that is your surface modification. And when you're already having the carboxyl group on your surface, that is surface passivation. So is it like I just gave an example? Is it more clear now?
Yeah, ma'am.
Okay. So moving ahead, carbon dots, they were accidentally discovered actually. Zhu et al. Basically in 2004, he was purifying single walled carbon nanotubes and he accidentally discovered carbon dots. So this discovery triggered extensive studies to exploit the fluorescence properties of carbon dots. Okay, so what are carbon dots? Carbon dots are biocompatible. They have small size with relatively large surface area. They are photostable and have photoluminescence properties that we have already seen that they are photoluminescence. So relatively large surface area, we know that more smaller size, more surface area. Okay, and they're also very photostable, like they're very stable photoluminescent molecules. Particles. So carbon dots, they're currently emerging as a class of promising fluorescent probe on account of their low photo bleaching, versatile surfaces, and excellent biocompatibility. So it's highly biocompatible. So instead of using quantum dots, you can use carbon dots for tumor imaging and all those biological purposes because it has excellent biocompatibility. Versatile surfaces means that it has so many different kinds, like I said, you can prepare anything with a carbon source, anything which has carbon in it, you can prepare carbon dots from it. So, so many kinds of sources you will have. So each kind of carbon dot will have different kind of surface functional groups. So versatile surfaces. Basically, that is what it means by versatile surfaces. And low photo bleaching means if you synthesize carbon dot, then it will, like I said, photo bleaching will happen, but it will be very, very slow, very, very slow. Fluorescent carbon dots have attracted increasing attention due to their potential application in sensing, catalysis, and biomedicine. Heteroatom doped carbon dots have enhanced fluorescent properties. So this is something which is very important. Heteroatom doped carbon dots. So basically, if you synthesize carbon dots from natural sources, like I said, it will have if you're synthesizing from plant extract, it will have some kind of natural stabilizers. So it can also have some natural dopants in your plant extract. So you do not have to dope your carbon dots. Usually what is meant by doping is that if your carbon dots that you have synthesized, if it has a very big band gap, then you need more energy to excite electrons from your conduction band from your valence band to conduction band. So what do this doping material do is that this doping material provide shells. I'll show you. Yeah, so this picture you can see, this is the conduction band and this is the valence band. So doping, what it will do is that it will give you instead of the conduction band here, you're using a dopant which provides you a shell below this. As a result, if you are exciting electrons here, maybe the band gap is not too high, but if it requires more energy to excite more, like the number of electrons you excite, more energy you need. So if your conduction, if you get an extra band here, level here, then more electrons will be excited to that level with less amount of energy. That is the work of your dopant. As a result, your quantum yield, the term that we use is called quantum yield. That is increased. Quantum yield is basically number of photons that is emitted divided by number of photons that were absorbed. So your fluorescence property of your carbon dot increases when you perform heteroatom doping. Usually you have to do doping if you're using some kind of chemicals for synthesis, then you have to perform some kind of doping. But certain times if you are doing green synthesis, then there could be some natural dopants and you might not need doping. But if you see that your quantum yield is less, basically after synthesizing your quantum dot, you quantum carbon dot, sorry, you have to do PL spectroscopy to find out its quantum yield. If you see that the quantum yield is less, You have to do doping to increase the quantum yield. Is it clear? Is it clear? What is doping? Why you need to do doping in carbon dots?
Yes, ma'am.
Okay. Another example also I can give you. Usually we perform this in lab for master student to show them how to synthesize carbon dot. We take a carbon source is basically your carbon source is citric acid. Okay, citric acid is your carbon source. The method that we use for synthesis is microwave assisted synthesis, and we use urea as a doping agent. So if we only synthesized using citric acid and microwave assisted synthesis, the quantum yield will be less. When we use urea, the quantum yield is more. So that is an example of a dopant I gave, which is urea, and your carbon source is citric acid. So urea will basically provide you nitrogen. It's known as nitrogen doping. Urea will provide you nitrogen groups in your carbon dots, so it's known as nitrogen doping. N doping. So you will see in papers it is written as N doped carbon dot or phosphate P doped carbon dot, like that they will mention. So that means they have used that material to as a doping agent. Okay, moving on to the lecture again. So synthesis. So there could be various methods for synthesis of carbon dots. You can use top-down synthesis methods like laser ablation, chemical oxidation, arc discharge, sonication, and electrochemical oxidation. But bottom-up approaches are mostly used for carbon dot synthesis, like thermal carbonization, which is thermal carbonization is basically pyrolysis. You take a sample and you just burn it. The ash that you get, that will have your carbon dots. You dissolve it in water, you throw the excess like whatever is big particles you have, your solution, whatever will dissolve in the solution is your carbon dot, and whatever remaining you have, that you have to centrifuge and just throw. And you have to filter, because there will be different sizes of carbon dots found, so you have to basically filter it so that you get a similar sized carbon dots. So that is thermal carbonization. Then we have microwave assisted synthesis. This is very common. Apart from pyrolysis, like these three are mostly used. One second. Okay, yes. Using thermal furnace. It's called muffle furnace. Yes. Yeah.
Yes, how can we differentiate the sizes in different groups? Like if
If
we want to collect 10 nanometer together, 30 nanometers differently,
Oh no,
is
no, you
that
cannot.
possible?
You cannot do that. You cannot.
Okay.
If you're thinking that in one setup you are synthesizing carbon dots, and then you will separate each size like that, that will be very difficult. What we usually do is that, like, you will see synthesis of yellow fluorescent carbon dots, red fluorescent carbon dot, orange green fluorescent carbon dot. So what they do is that they use different reaction times, what temperature and what how long you're keeping in your keeping in the furnace. That is very important. So each condition will give you different sizes of carbon dots. If you keep it for a longer duration, mostly the most common carbon dot you get is the blue emission carbon dot, which is the smallest size. But if you want bigger sized particles, then you have to change reaction conditions. Sometimes you can also change your doping agent as well. So like that you can do some kind of tweaking, but at a time you will get only one particular fluorescent carbon dots. You cannot get multiple fluorescent. So because if you have different sizes, like when I am saying different sizes, it does not mean that very variation in sizes. Maybe all are emitting blue light, all are emitting blue fluorescence, but the sizes are different, varying. Maybe there are some macro particles, so that is why we filter it out and keep the almost similar size particles together. But it will not be like some red fluorescent, some yellow fluorescent. They will be mixed with your blue fluorescent carbon dots. At a time only one fluorescent kind of carbon dot will be formed.
Okay. And in what for the bottom up approach,
Yeah.
in what type of solution do we dissolve that?
Bottom up approach, it depends. Suppose, like I said, the source. Like I'm giving an example, like I synthesized carbon dot from a plant source. I have taken a plant leaf. So what
Mhm.
I did was I took the plant leaf, I washed the leaf. You have to clean it very nicely. And what you did, I will come to this thermal furnace that you mentioned. I was coming to that bottom up approach. That is most commonly you will find in labs that they use. It's called the hydrothermal method of synthesis. So for that you have a small autoclave. So I'll just share the picture. Can you see the PPT? Can you see the
Yes.
PPT? Yeah. So you can see this small autoclave that you can see here. Not
Yes.
autoclave. This is like a small reactor basically.
Mm.
So this reactor, in this reactor you will put your material from basically your carbon source, and we usually put water. So we will cut the leaf into very small pieces, and then we will put it in water, and we will put some water, and we will put it in this reactor. And then there's an autoclave. We will put it in the autoclave, set the temperature, and we will set the time also. Stepwise the temperature will increase, and the reaction will go on for maybe 6 hours, 10 hours, depending on your requirement, depending on the literature survey you have done. You're getting the protocol maybe from some paper, so you will have a protocol already what they have done to synthesize that particular carbon dot. And then you can optimize it by yourself, because there will be some differences, so you have to optimize it a little bit to get your exact requirement. Okay, so microwave assisted synthesis is very easy. You add it in water. You take your like I said, like I was giving the example, citric acid. You take citric acid, dissolve it in water, little bit amount of water, and you basically put it in a microwave, heat it till the water is boiling, then take it out, cool it for a minute, then again put it, heat it till it's boiling, again cool it. Like this you have to do till your solution turns brown, till the water completely evaporates and your solution starts to burn and it starts to become brown. But you have to be very careful so that you do not light a fire in the microwave. Okay, then you have to add the water and dissolve the carbon dots that are formed. So that is the microwave assisted synthesis. Whereas the hydrothermal one is even easier. You do not have to do anything. Just after setting it up, you just put it in the this reactor and just keep it in the autoclave. Only you have to switch off the autoclave at a particular time that you have to remember. You have to note it down, and that's it. And after that, you just take it, purify your carbon dot, and then you can visualize under UV to see whether it is giving the fluorescence or not. Is it clear?
Yeah, I was also talking about the thermal carbonization, which you
Yeah.
told
Yeah.
that it is just a ash.
Yeah.
You take the ash and you dissolve in
Yeah.
water.
Yeah. No, see, this is the
It is
hydrothermal synthesis.
deionized, or it is deionized water, or it is
Deionized water.
simply
Deionized water. Whenever you're
okay.
doing nanoparticle synthesis, everything should be done in deionized, milli-Q.
Mhm.
Everything should be done in deionized water. So this is the reactor I was saying. So what this is basically, can you see this white container? This is the chamber, hydrothermal synthesis reactor chamber body. So this chamber body, in this chamber body, you will put your sample dissolved in your solvent. Okay, we do not usually we put water. We do not put any flammable object or anything, because your autoclave runs at very high temperature. It will run at 160 degrees or 200 degrees. Uh, Usually this chamber body. One second, somebody. Okay. Usually, this chamber body comes with various sizes and temperature. Like it can withstand a particular temperature. So, like the one which we have in our lab can withstand only 200 degrees. So you should use it less than 200 degrees, not even 200 degrees. It will melt. Basically, it starts to melt. So this is your cap. You have to put your sample with mixed with water. Now plant leaf will not dissolve in water. You're just cutting the plant leaf in small pieces. You're putting some amount of water, volume of water, and you're putting the plant leaf in it, which you've cleaned thoroughly, cut into small pieces because your entire leaf will not fit into it. And then you're setting this entire thing up and putting in the autoclave machine and keeping it. After it comes out, you will see that the water itself will have some brownish tinge to it. So basically, you do not need the ash. You just need that brownish solution. Your brownish solution will contain your carbon dots. So, but what we usually do is we will take the entire content in a falcon tube and we will centrifuge it, so that in case your water also contains bigger particles, that will settle down and only you get a very uniform solution. Is it clear?
Yes.
Okay, so I will tell you in a little short brief that how you can set this reactor up. Very important, because if you do not set it up or you miss one part, it will like damage this chamber body, which is very crucial and very expensive. It's like 11,000 or 15,000 one chamber. So and this outside body is even more expensive. It's 25,000. So you should not lose any part. So basically, this chamber will have a bottom disc. The bottom, I think the bottom disc is not shown here. This small circular silver colored dish that you can see. So this kind of dish will one will be disc will be going at the bottom. You put it at the bottom. Then you put your chamber body. You cap your chamber body. This disc, another one you have to put on top. You should never forget to put this disc on top, one at the bottom and one at the top. You should never forget, or else when you're keeping it in your autoclave machine, it will melt your chamber body. So two discs, one at the bottom, then your chamber with your sample, then the top, then you put the lid on, and this is the rod. Basically, you have to tighten it. So when you're using your hand to tighten it, you will see that you will feel that it is tight, but when you put the rod in and you rotate it, you will see that it is tightening more. So this should be completely screwed shut, and then you have to put it in the autoclave machine for hydrothermal synthesis. So depending on your reaction conditions, like what temperature and what is the duration you are keeping your sample for, you will get different sizes of carbon dots, and each one will give you different fluorescence. Is it clear?
Yes.
Okay. Any other queries?
Yes, I wanted to know regarding that I am still stuck with that.
Peptide ink?
No, not that. Please show that slide earlier.
Which slide?
Uh, here, thermal carbonization.
Yeah.
You told that it will give a fluorescence. Like once we dissolve
Yeah.
and we centrifuge and we take out the macroparticles,
Yeah.
then what would be the next step?
Basically, sometimes we filter it, syringe filter it, 0.22 micron filter. We will syringe
Okay.
filter it, and then we will just visualize it under UV. Basically,
Okay.
like filtering should be like, when you're filtering, you should always keep a track that your solution is not becoming clear, like you're not getting clean water. Okay. Usually the
Okay.
0.22 micron filter will not retain carbon dots, but sometimes something might happen. Maybe something might be wrong with the filter, and it can happen, because it has happened to me once. Even though those filters do not retain any kind of nanoparticle, but I think there was some problem with the filter. It retained my gold nanoparticles, which was around 10 to 14 nanometer range. which was very unusual because the filters do not retain nanoparticles. But some kind of maybe problem was there in that batch of filters. So you should always, like whenever you're filtering, you should notice the filtrate that is coming out, whether you're getting clean water or not. If you're getting clean water, you should immediately stop filtering it, and you can just use it you have already centrifuged it, so means all the extra particles and macro particles are settled. So then you can directly, if needed, you can perform one dialysis also. That also you can do dialysis. Instead of filtering, you can do dialysis so that bigger particles leave and you only get the small particles. But every time you have to discard the water very carefully, because that will contain your fluorescent nanoparticles, like some kind of carbon dots will be there. So you have to like discard it very properly and clean it very properly. So that is
So
your basic.
with the help of dialysis, what is the range of the nanoparticles we retain?
That will depend on your dialysis membrane. Usually, the smaller sized particles remain inside, the bigger ones go outside. But that
Yes.
will depend on your dialysis membrane pore size.
Give two examples. Like if the dialysis pore size is this much, then you retain this much size.
Usually, we do not like carbon dots. Will not be more than 10 nanometer. Right.
Okay.
So any kind of carbon dot, it will be retained in your dialysis membrane.
Okay.
What dialysis
Okay.
membrane we have used is it was 10,000 molecular weight cutoff.
Okay.
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 So we used a 10,000 molecular weight cutoff dialysis membrane, and our carbon dots were retained. It was not like there was too much loss.
If at all, if we want to retain gold nanoparticles of different sizes, then
Same. You can use the 10,000 molecular. In my lab, we use the 10,000 molecular weight dialysis membrane.
and okay, so in that case, how what size of nanometer we retain?
Nanometer size usually up to, I think, how much you can say. Like, I think I have usually worked till 20 nanometer, not more than that. 20,
Okay,
30,
till
30
20
nanometer.
nanometer we retain
No,
the
that is
sizes.
no.
Okay.
That is my personal experience. Like, I have only done
Okay.
till that much. Maybe it can retain higher than that.
Okay.
Okay.
Fine. Thank you.
Because 10,000 molecular weight, that means you can understand it is for protein, retaining protein. Proteins are not less than 100 nanometer. They're always bigger
Okay.
than 100 nanometer.
Okay.
Even though proteins are
Fine.
considered as nanomaterials, it's a 0D nanomaterial, but proteins will be, depending on what kind of protein you choose, it will be big in size. So 10,000 molecular, 10,000 molecular weight means it should be able to retain most type of nanoparticles without loss.
Okay. Fine. So is there so through dialysis only we can retain different sizes, right?
But dialysis is one option. Like I said, you can do
Okay.
filter also. Like I experienced it. Like in my filter, the gold nanoparticles were getting stuck. So that time the option is dialysis. Dialysis mainly, actually, you do not only to remove big particles. Mainly, it is done to remove your extra reactants. For in the case of carbon dots, you do not have any extra reactant. You're just adding water. You're adding your sample, and you're just burning your sample. So you're carbonizing it basically. So you do not have extra reactants unless you're using some doping agent extra. So those are your extra reagents. Like when you're doing metal nanoparticle synthesis and you have excessive of chemicals left behind, like excesses of excess of stabilizers. Although you need to have perfect concentration, then only you will get a good stable nanoparticle. Too much of stabilizers, your particles will aggregate. Too less stabilizers also, it will aggregate. But sometimes unreacted stabilizers can remain, and sometimes when you're using reducing agent which are toxic, like NaBH4, sodium borohydride, when you're using such toxic reagents, so it is important to remove them before you're going for any kind of biological application, at least. So that time we do the dialysis, so that slowly that excess reagent, if it is there, then it will be removed.
Sonication can give us what size of nanometers?
Sonication, again, it will be depending on what kind of how much time you're sonicating. Like everything depends on your reaction parameters.
Timings.
Yeah, timing.
Parameters.
You
Yeah.
cannot ask like what kind of nanometer range you will get. It everything depends on
Okay.
your experimental setup. You cannot.
Setup. Okay.
No, it's not like
Yeah.
one particular technique will give you only this sized. That is not.
Okay.
That is why the optimization. That is why you have to perform
Okay.
so many experiments. You have to perform with so many different conditions to see which one is best for your work.
Okay. Fine.
Okay.
Thank you.
Okay. Okay. Uh, So this is the hydrothermal synthesis I already explained, and this is the application of CDs in therapy. So basically, what you are seeing here is you have the cancer cell, and you have the cancer cell membrane. In the membrane, you can cancer cell membrane, and this carbon dot when this PNN basically is a semiconductor term, P for positive and N for negative type carbon dot. So basically they have mixed two different kinds of carbon dots, one is P type, one is N type, and formed a combination of the both of them, PN carbon dot. And then when this carbon dot enters your cancer cell, like a cancer cell membrane, you're packaging your carbon dots inside this cancer cell membrane so that it is taken up by the cancer cell. Is it clear? As you can see here, this cancer cell membrane is being taken up by your cancer cell. So when you're injecting this solution in your mouse which is having cancer, I think in this case it was liver cancer. This mouse had liver cancer basically, and the kind of therapy it was going for is sonodynamic therapy. So sonodynamic therapy is basically you are using you're using some kind of waves to generate ROS. So what is happening is that this whenever you are using waves, you can see here that your GSH is depleted. So what is happening is your GSH will be depleted, and this energy is transferred to the electron which will get excited. So this is how your carbon dot will work. Your GSH, from GSH, it will take the energy and will be excited to the higher level, and it will go undergo intersystem crossing, and you can see here singlet oxygen, that is ROS, being formed. So your GSH is being depleted and your ROS is increasing. So this will provide oxidative stress to the cell, which will cause oxidative damage, and the cell will die. So this is how you're targeting only cancer cells to die for therapy purposes. So as you can see here that before they injected the mice with the before they injected the mice with the carbon dots, you can see how much the tumor is, and after injecting you can see how less the tumor has become. Did you understand the application of how it is used for therapy? Any questions? Okay. So, in brief, we'll be talking a little bit about photoluminescence because this is very important when you talk about fluorescent molecules. So, what is photoluminescence? So, photoluminescence is basically the phenomena of temporarily light absorption and subsequent light emission. It's called photoluminescence. So, here you can see this is absorbance, which is your UV. UV gives your absorbance, and then PL will give you fluorescence and phosphorescence. Uh, Photoluminescence, which occurs by virtue of electromagnetic radiation falling on matter, may range from visible light through UV, then X-ray and gamma rays. Okay, so photoluminescence is a process in which a molecule absorbs a photon in the visible region, exciting one of its electrons to a higher electronic state, electronic excited state and then radiates a photon as the electron returns to a lower energy state. So usually the photon could also be absorbed in the UV range. So maybe the your material will absorb photons in the UV range and then radiate a photon in the visible range. That is how you're seeing the fluorescence. Okay, so this is the phenomena of photoluminescence. So what happens in the PL process is basically three main processes. One is excitation, the other is relaxation, and the last one is emission. So here you can see your electrons are there in your valence band. You're providing energy. So your electron will only absorb that photon energy which has energy equivalent to your difference between the valence band and the conduction band. Okay, if your photon has energy either equal to or more than the difference between the valence band and the conduction band gap energy, then only your electron will be excited to higher energy states. And non-radiative relaxation is basically before your electron comes back to the ground state, it will like use some of its energy, excited state energy, to come back to the lowest level of the conduction band. So this is non-radiative relaxation. So no kind of photon is released when it is coming down. So basically it is using some of the energy. So when it is getting excited, it has an energy h mu 1, but when it comes down, it has a different energy h mu 2. That is why you will see a shift in your excitation peak and your emission peak. Do you know what is the shift called? Do you know what is the shift called? Your excitation peak and emission peak, there's a difference between your excitation peak and the emission peak, emission spectra. So, do you know why that shift happens? I just gave an explanation why that shift happens. So basically, this non-radiative relaxation uses some of the energy. So this green energy and this red energy is not same. This one is higher, this is lower. So where will it shift? It will red shift. Right? When you see the electromagnetic spectrum, UV wavelength is low, energy is high. And as you go down the spectrum, your wavelength is high, but energy is low. So it will become red shifted. Your spectra is red shifted. So you can see here, this blue line here shows your excitation spectra or absorption spectra. You should know that absorption and excitation spectra are same. So if you suppose you have taken a carbon dot, instead of using PL, you are doing UV visible. And you are finding out what is the absorption of your carbon dot. So wherever your carbon dot absorbs, be it in UV or visible, wherever range, usually carbon dots will absorb in UV range. So if you get a peak in the UV range, that peak is your excitation for your carbon dot. So you need not find out the excitation peak again in while doing PL. You can directly use that absorption maxima and get your emission spectra. But it will always be red shifted. As you can see, it is moving towards the red spectrum. In the visible range, 400 to 800 is the visible range. So it is shifting towards the red. So this is known as red shift. So this shift, you can see the difference between the two peaks, the absorption spectra or excitation spectra and the emission spectra. This shift is known as Stokes shift. You will always observe this Stokes shift. So that is why what we do is when we are doing when we are finding out the excitation spectrum of a carbon dot or any fluorescent molecule, you will be scanning it from 200 to 800, or maybe even in the NIR. If you have NIR range also, you can do. Usually we do from 200 to 800. So wherever you get the peak, whenever you are doing emission spectra, you are always supposed to start, like you're putting the excitation spectra you're getting a excitation spectra value, you are supposed to start 20 to 50 nanometers ahead from the excitation spectra value to collect your emission spectra, or else you will get error in your graph. So this is a thing you should always remember when you're doing PL spectroscopy. You should, if your suppose your excitation spectrum is 320, here I think it is somewhere around 480. So if your excitation spectra is 480 or 490, you're supposed to start collecting your emission spectra value 20, at least minimum of 20 nanometers ahead of your excitation spectra peak. So if it is 490, you should start collecting from 510 or 520 nanometers. Okay, this is the graph just for representation. But basically you have to collect it like that, or else you will get some errors in your result. You will get some extra peaks down, which can mislead you. So the photo excitation causes the material to jump to a higher electronic state and will then release energy. So photons, as it relaxes and returns to a lower energy level, the emission of light or luminescence through this process is called photoluminescence. So basically, you have an electron in this S0, which is the ground state. You are exciting it with energy, which is equivalent to the energy difference between S0 and S1, or either it is same or more. If it is going to S2, then there will be some non-radiative relaxation and it will come down first to S1. Then there will be a radiative emission, that is E emission. This you will get as a peak. This non-radiative emission you cannot find. This is the energy loss that happens. That's as a result, this Stokes shift is observed. Any questions? Okay. So PL process. So PL is a process in which a molecule absorbs a photon in the visible region, exciting one of its electrons to a higher electronic excited state, and then radiates the photon as the electron returns to a lower energy state, because excited states are unstable. If the molecule undergoes internal energy redistribution after the initial photon absorption, the radiated photon is of longer wavelength, that is lower energy than the absorbed photon. So like I said, it will be red shifted. So it is absorbing in the green region, but when you are getting an emission band, it will be red shifted, longer wavelength and lower energy. Okay. So this is the Jablonski diagram. This is very important to understand PL. So basically you have a band gap and you are exciting electrons from a low ground state to higher state. When it absorbs photon, then there will be some internal conversion. If it goes to very high states, there will be some internal conversion and it will come down to the lowest singlet excited state. Okay, lowest singlet excited state. It will come down first. Then there could be different phenomena. One could be quenching, where you will not get any emission. There could be internal conversion, or again non-radiative decay, where your electron will just come down without any emission. And next one is fluorescence. In this fluorescence thing, you will get a photon, emission of a photon, but with different energy. The excitation photon had more energy and the emission photon will have less energy. And another thing that can happen is intersystem crossing. While your electron is in the excited S1 state, it can go intersystem crossing and it can go to the excited triplet state. So what is the difference between the singlet state and the triplet state is, electrons will have two different spin. One is up, one is down. So when your electron, so out of the electron pair, up spin and down spin, so only one will be excited. When you have fluorescence, then you have, suppose the ground state one is up spin and the down spin one is excited to your S1 state. So it is of opposite spin. So if that opposite spin electron directly comes down, it will give fluorescence. But if that opposite spin goes intersystem crossing, somehow it will change spin and become the same spin as the ground state electron pair. Okay, so if this is up spin, this will also become up spin in triplet state. Okay, and after that it will again change spin to down and then come back to the ground state. So this entire process, where you can see from ground state it is going to singlet state, then undergoing intersystem crossing to the excited triplet state, and then coming down back to the ground state, there will be a different kind of emission. This is known as phosphorescence. So this will be more shifted. This will have even less energy compared to fluorescence. This will be more shifted towards longer wavelength, and it will also take more time. So fluorescence and phosphorescence is, fluorescence will, you might get it immediately. Phosphorescence is known as delayed fluorescence. So it takes some time because the electron is undergoing so many processes, first excitation, then intersystem crossing, then it will change spin and come back. So this entire process takes a little bit more time compared to fluorescence. So that's why it is called delayed fluorescence or phosphorescence. Okay, so this is a difference between your fluorescence and phosphorescence. So fluorescence is spontaneous emission of electromagnetic radiation, whereas phosphorescence is delayed emission of electromagnetic radiation. Fluorescence will be glow of fluorescence stops right after the source of excitatory radiation is switched off. So as soon as you switch off your radiation source, the fluorescent molecule, suppose you, like if you see carbon dots under UV, you switch on the UV, you see the fluorescence, you switch it off, it will immediately switch off. You will not see any kind of fluorescence. Okay, so that is fluorescence. But in phosphorescence, what happens? Even after switching off your UV source, you will still see an afterglow. Okay, so phosphorescence takes time for emission, and it will also be retained even after for some durations, maybe up some fractions of a second or up to hours, depending on the material. So it will take some time to die out. Okay, first fluorescence is switch on, switch off very quickly. Phosphorescence will take time to die out. Another difference is light production by the absorption of UV light, resulting in immediate emission of visible light. This is in case of fluorescence, like I said, giving the example of carbon dots. And phosphorescence is light production by the absorption of UV light, resulting in emission of visible light over an extended period of time. This is the afterglow that they were talking about. So fluorescence is from ground state, it will go to excited singlet state and then it will come back to the ground state. Phosphorescence from ground state, it will go to this excited singlet state. Then it will undergo intersystem crossing to the triplet state and then go back to the ground state. Okay, some example of fluorescence is highlighters, pens, fluorescent lighting, fluorescent dyes in detergent. Whereas for phosphorescence is any object which you coat with phosphors. So phosphors basically give you this phosphorescence phenomena. Any questions? Any questions? Okay. Okay. So PL spectroscopy. Now we come to the spectroscopy. So these are certain facts that you should remember about PL spectroscopy. So PL spectroscopy is a non-destructive spectroscopic technique commonly used for the study of intrinsic and extrinsic properties of both bulk semiconductors and nanostructures. So PL spectroscopy is when light energy or photons stimulate the emission of a photon from any matter. PL spectroscopy is a contactless, versatile, non-destructive, powerful optical method of probing the electronic structure of materials. The intensity and spectral content of this photoluminescence is a direct measure of various important material properties. It gives information only on the low-lying energy levels of the investigated system. Okay, so it can only give you information about vibrational levels, but as you can see in the Jablonski diagram here, you can see vibrational energy states, but you cannot see there are even more states in between. Those states you cannot, those are rotational energy levels, those you cannot get through PL spectroscopy. So during a PL spectroscopy experiment, excitation is provided by laser light with an energy much larger than the optical band gap. Like I said, so this band gap, your laser light should provide more energy compared to this band gap. Then only it will excite electrons. So the photoexcited carriers consist of electrons and holes which relax together toward their respective band edges and recombine by emitting light at the energy of the band gap. So when you are exciting an electron to a higher band gap, when you are exciting an electron to the conduction band, the absence of the electron in the valence band is called a hole. So though both of them will try to recombine, they will come back to their band edges. Band edges basically means that coming to the lowest singlet excited state, it will come to the edges and then it will try to recombine by emitting light at the energy of the band gap. So the quantity of the emitted light is related to the relative contribution of the radiative process. So depending on how the electron, what kind of processes it goes through, how much it has been excited, how much energy it has to spend in coming down to this lowest state, everything, all of these factors will be combined together to find out that what will be your emitted energy of your emitted light. So radiative transitions in semiconductors may also involve localized defects or impurity levels. Therefore, the analysis of the PL spectrum leads to the identification of specific defects or impurities, and the magnitude of the PL signal allows determining their concentration. Okay, so if you have some defects in your material or you have some impurity, so all of this you can use PL spectrum, because each impurity and defect will give you different kind of spectrum. So you'll be able to identify how much your spectrum is, how much defect is there and all, and how much impurity level is there. So all these you can find out through the PL spectrum. The respective rates of radiative and non-radiative recombination can be estimated from a careful analysis of the temperature variation of the PL intensity and PL decay time. And at higher temperatures, non-radiative recombination channels are activated and the PL intensity decreases exponentially. So this is a thing you should remember. Like if you're using the PL instrument for a long time, it tends to heat up. Any instrument or any gadget you use for a long time, it will tend to heat up. So if you can understand your instrument is heating up, you can see here it is written that with higher temperatures, non-radiative recombination channels are activated. So this kind of non-radiative relaxations will happen more compared to your radiative relaxations. So as a result, you will get less intensity. So intensity will decrease exponentially. So whenever you see your instrument is becoming a little bit heated, you're supposed to switch off the instrument, let it cool for some time, let it come down to room temperature, and then again use it, so that you do not get any decrease in intensity while doing PL spectroscopy. Okay, so this is the experimental setup for your PL spectroscopy. So this will be a light source. Do you have any idea what is the light source for PL spectrum? Can any of you tell me which is the light source for PL spectrum? Light source for UV visible? Light source for UV visible? No, not UV light. No, it's not UV light. You have lamp, some material lamp. What is the material? You have two lamps in UV. You should remember, one is a tungsten lamp, another is a deuterium lamp. One tungsten for, as far as I remember, it is for visible light, and deuterium for UV. Okay, two lamps in UV visible, one for UV range, one for visible range. Please confirm which lamp is for which range, tungsten lamp and deuterium lamp. For PL spectroscopy, you have one lamp only, which is xenon arc lamp. So xenon arc lamp basically for your PL spectroscopy. So the difference between your PL spectroscopy and UV visible is that UV visible as you can see, this light source is your xenon arc lamp. You will get multiple wavelengths. So you have a monochromator, which is called the excitation monochromator. So when you are putting a wavelength in the software, whenever you're putting the wavelength, excitation wavelength, so this monochromator will only allow light of that wavelength to pass through. Okay, so that is your excitation, lambda excitation, one wavelength. It will go to your sample cell. Your sample cell will have your sample, which should not be too much concentrated. If it is too much concentrated, then it will have an effect called inner filter effect, where the wavelength will be like scattered more compared to absorption by the sample. Compared to the sample absorbing the wavelength, it will be more scattered by the molecules. So your sample, whenever you're preparing sample, it should not be too much dilute, it should not be too concentrated. You have to maintain that so that you avoid the inner filter effect. Then what is happening is you will get luminescence at various wavelengths, but your emission monochromator will only select one wavelength and let it pass, which will be detected in your software, which is connected to a computer. So you'll get the emission spectra. So the difference of UV visible and PL spectroscopy is, firstly, UV visible measures transmittance, and here you are measuring emission. Second is UV visible, the detector is at 180 degrees, like straight, it's in one line. This will be a light source, here will be your detector for UV visible. Whereas for PL spectroscopy, you can see your light source is here and your detector is at 90 degree. So this 90-degree angle is basically because when your wavelength is being excited, when the wavelength falls on your sample, transmitted for UV visible, it is at 180 degree, because transmitted light is always at 180 degree. Your instrument will detect the transmitted light, use the Lambert-Beard law to find out the absorbance. Okay, so basically UV visible measures transmitted light, not absorbance. It will use a formula to convert the transmitted light to absorbance and give you the absorbance value. Whereas for your PL spectroscopy, this you have fluorescence. Fluorescence, unlike transmittance, is not in one direction. It will be in all directions. So you can put the detector anywhere. But if you put it at 180 degree, apart from your sample, there will be other molecules present in the solvent. The solvent will also have some molecules, and there will be some scattering also by your samples. So that scattered light will be here. The some will be transmitted light at 180 degree, some will be scattered light here and there, and that scattering is least at 90 degree. Scattering of light by your sample or solvent will be least at 90 degree. So that is why the detector is kept at 90 degree for your PL spectroscopy. Okay, is it clear? Any question? Okay, any questions, any queries you have? Okay, so we come to today's end of the session. So last day, I think the one ma'am, like she asked a question, application of peptide ink. So as you can see, I have shown here. Ma'am, can you see?
Yes.
Yeah.
Yes.
Okay, okay.
Yes, it's visible.
So application of peptide. Shall I explain? Okay, first let me tell you the application, then I'll explain to you that peptiding thing. I think certain questions you asked me. First was what is the application of peptiding? What was the why you're using only hydrophobic amino acids in the linker region?
Linker. Yes.
Yeah.
Yes.
Another thing you asked me that how are cells adhering?
Yes.
Yeah.
Yes. Yes.
Okay. How are cells adhering? Regarding that Cephardex thing you asked me, I said that it is basically in column chromatography also it is used. So to be more precise, Cephardex is used for sample purifying sample. It is used for desalting samples. Okay, in column
Desalting
chromatography,
the samples.
desalting
Okay.
samples, yes. So to remove salts from your sample, that Cephadex column is used. You run it through a Cephadex column in column chromatography. So that is why if you search also about Cephadex, you will get different sizes of Cephadex. It will come like it is marketed in different name.
Okay, fine.
Okay,
Thank you.
so that is the exact work of your Cephadex, and regarding that cationic DNA thing, I'm sorry I did not find it in any module. I think
Okay.
is it like solid lipid nanoparticle or something like that?
Uh, 1 second, I'll just find it where I've written it. The cellular uptake liposomes, the module in which the liposomes are explained.
Okay, I'll open that PPT. You can tell me which one. Then maybe it will help me also identify. Maybe you could not exactly
Yeah.
pinpoint it.
Yeah, yeah.
So let's just first talk about
Yes.
the application of peptide ink. So
Yes.
application of peptide ink, as you can see, basically it's a ink means it's for 3D printing. Whenever you are making a ink, it's for 3D printing. I think has 3D printing been taught to you yet, or it will be I think in the last week course.
No, 3D printing is not yet. I have not
Cell
come across it.
organ printing.
Organ printing is not done. Someone
Okay,
else can
okay,
just
okay,
guide if they have done.
okay. So maybe organ printing. 1 second, I'll just check if anybody said anything. Okay, so organ printing is basically okay. Unit 3. Oh, week 3, it has been done. Okay. Thank you so much. So, in organ printing, you might have heard about different kind of printer, laser printer, inkjet printers. So, 3D printing. So 3D printing is basically so this peptide ink that you're making, this is basically used as a bio ink for 3D
Mm-hmm.
printing. Okay, so this ink could be in the form of, you can see this peptides, you can make it in the form of fibers, nanotubes, nanovesicles, anything. So, use it as an ink, and you can see various applications. It like tissue engineering, drug delivery, bioimaging, diagnostic, and organ transplant. Basically, if you're using for 3D printing to form organs, so it will be like the reason you asked that you're growing cells, what is the need to make a peptide ink and grow cells? It's basically like you can make skin grafts. Okay,
Mm-hmm.
so for burn victims, you can use the skin grafts. You can take some cells from other part of their body so that it's biocompatible to their, like there is no immune host graft rejection, and then instead of cutting a piece from other part of the body and putting it, you can do this 3D printing. You can make a peptide ink. You can grow that person's cells on the peptide ink, make a skin graft, and then you can put it back in the person. Okay,
Okay.
so that will come under tissue engineering as well as your organ transplant. So that is the basic use of your peptide ink. So this is also a very like I found it is a very innovative topic. A lot of but there are a lot of challenges. Like skin is relatively easy to make and graft, but organs like more highly vasculated organs like heart and kidneys and all, they are very difficult to form. So highly vasculated organs like heart are very difficult to 3D fabricated. So till now a lot of, yeah, any question?
Sorry, my child interrupted.
Oh,
Sorry.
okay. Sorry, no problem. Okay, so so basically we know that organ transplantation demand is too much. So currently researchers, they are developing different kinds of methods so that we can 3D fabricate organs, and then we can use them as or for organ transplant. And for this 3D fabrication, the best part is that there will be no host graft rejection, because you will be using the host cells to grow. Okay, so
Yes.
that is the application I found for your peptide ink. And just for your own knowledge, I included this protein ink also. So peptide ink is, protein is made up of peptides. So basically, you are just using some peptides to like form the scaffold and grow the cells. But you can also do this protein ink. So here you can see in this picture that proteins like collagen, gelatin, fibrinogen, elastin, then silk fibrin, fibrin, keratin, egg white, soy protein, milk whey. So these are like amino acid sources where various from human building block are amino acids, and from these amino acids you're getting these various kinds of protein. So these proteins, you will make protein based inks, and you will use them for sol gel transition by cross linking them and assembling them. You can see here they're 3D printing it. Okay, and various applications like tissue scaffolds, clinical implants, disease models, bioelectrons, drug delivery. So you can use it for a, since these are biocompatible, so you can understand these are protein. So if you like put it in your body for drug delivery, so your own body system will use this like break down these proteins and use the amino acids, as well as the drug will also be delivered.
Okay.
Okay, so this is also I found out one application of protein ink. Okay, so thank you today, and I will be discussing that. I'm sorry, 1 second. I will be discussing this. Can you see this?
Yes.
Okay. Uh, So basically, you asked me about that peptiding, the three regions. Like yesterday, I was mentioning that it should have three regions: ligand, anchor, linker. So you asked
Linker.
me how
Yes.
you understood how the peptiding is anchoring, right? Why the anchor is used?
No.
You did not understand.
No.
Okay.
No.
Okay.
No.
Okay. So basically, you can see here it is written that these peptides have three general regions along their lengths: a ligand for specific cell recognition and attachment. Okay. So ligand basically you're using for cell recognition and attachment. So as you can see here,
Attachment.
an example is given. RGD is a motif that is
Hmm.
known to promote cell adhesion. So you can understand how.
Yes.
Yesterday I told you that the ligands basically, if you have peptides, when you're using peptides, those peptides will bind. Every cell will have some kind of receptor for binding some peptides. So those
Hmm.
receptor will bind, the cell surface receptors will bind to the peptide and it will adhere. I told you yesterday,
Yes.
you remember?
Yes.
So to
Yes,
be
sir.
more precise, when you're using a ligand like RGD, which is basically arginine, glycine, and aspartic acid, this RGD motif is recognized by integrins on the cell surface, or ECM.
Hmm.
Okay, so it will bind to the integrins and it will help in cell adhesion. Okay, and after the cells adhere, we know that mammalian cells, they need to adhere in order to grow. Unlike cells, they don't need to adhere. They can multiply, but mammalian cells, they need to adhere first to a substrate and then grow. Regarding
Okay.
the linker, that why you are using a string of hydrophobic amino acids, this I did not find out anywhere, but possibly because I think maybe you need to, from the substrate, maybe you need to create some space in order for the cells to grow. So for that, hydrophobic amino acids are a little bit bulky groups. So maybe that is why they're using the linker to separate the anchor and ligand enough.
Okay. Fine.
Okay, so maybe that is why they're using the hydrophobic amino acids. That could be a possibility. And regarding the anchor, so you have the peptide. You can understand the peptide ink first has to adhere to the surface. Then only your ligand, when it is attaching to the cells, it will adhere. So for that anchor, you can see cysteine residues. So cysteine has SH group, right? So gold surfaces are very well known. Like you can modify gold surfaces with sulfhydryl groups, and with cysteine has sulfhydryl groups, so it will form disulfide bond with gold surfaces, which will help your peptiding to anchor. Okay, did you understand? You can modify gold surfaces with sulfhydryl groups, and then you can, with the cysteine residue and the sulfhydryl groups on your gold surfaces, will form a disulfide bond and it will anchor your peptide ink. Did you understand?
Not much, but I'll try to understand. It's okay.
Okay.
I'll go through the material, no
Okay.
problem.
Okay. Like, where did you not understand? Please tell me so that I will tell you again.
Uh, Actually, we are not much accustomed with this terminologies and everything. तो बीच
Okay.
में
Okay.
link छूट जाता है तो
Acha, acha. So which terminology you did not understand? Please tell me. I will tell you.
नहीं ऐसा नहीं है बट लाइक थ्रू आउट द फ्लो तो बीच में कहाँ छूट गया है मुझे भी नहीं पता है आई वाज
Acha,
लाइक
okay, okay. So, to be more precise, gold, I said, when you're preparing gold nanoparticles, to make it more easier, suppose you have a stabilizer which has sulfhydryl group.
ओके
Okay, maybe you're preparing a gold nanoparticle with cysteine only as a stabilizer. Okay, so your
ओके
gold surface is having cysteine, so it will have, if you
यस
see the cysteine residue amino acid picture, you will see it has an SH group.
ओके
Okay, so that is called the sulfhydryl group. So if your
ओके
anchor also has a cysteine group, then both are having SH, and in proteins we know protein cysteine forms a disulfide bond, SS bond, right?
यस
So your gold, this anchor, if your gold, suppose you have to make it on a platform. If your platform is gold, then you can functionalize with cysteine. Like your anchor can have cysteine residues, your gold surface should have an SH group so that it can form a disulfide bond and anchor your peptide ink to the surface. Okay,
ओके
so if your peptiding
गॉट इट
attaches to, so anchor is basically to anchor, to attach your peptiding to the surface substrate.
सर
Okay,
ओके
and then
या
the linker to create a space between your anchor and the ligand, and then
ligand
ligand will bind your cell for cell growth.
cell growth ओके
Okay,
गॉट
okay,
इट या
so you can have different kind of surfaces, not necessarily it could be gold surfaces. So if you have suppose amine surfaces with amines in it, remember we were talking about EDC NHS chemistry.
यस यस
So basically in EDC NHS chemistry, it was forming what I said, a amide bond. So aspartic acid will have a COOH group, and your substrate, if it has amine, then it will also form a amide linkage, CONH2. Similarly, lysine will have amine and carboxylic surfaces will have COOH. Again, it will form an amide bond and it will anchor your peptide ink.
peptiding okay
Okay. Any other confusion?
no no
Is it clear, or should I give any
yes
more
no
easier
no
example?
now yeah this process is clear
Okay.
that how it forms that a base has been given
Yeah.
then it has an anchor which
Yeah.
adheres to the base
Yeah.
then there is a linker which creates the gap between the anchor and the ligand
Yeah.
and the ligand then attaches to the cell for
Yeah.
the cell growth
Yeah. And
yeah this
this
much is clear
peptide
now
ink basically, it will help in, drawing a pattern. So if you
yeah
put,
वो नहीं समझ में आ रहा है that I'm not able to
ha,
understand
so basically what will happen is that this peptide ink, you will, as you can see here, it is written MIT. So this MIT basically is cells growing on your peptide ink. Okay, so
okay
instead of making a proper mask, suppose you are 3D printing some organ, so you have to have a mask that this organ should be printed in this shape. So you do
okay
not need to, but that mask, there will be a lot of problems. You have to separate the mask, you have to do something. Here in this case, your peptide ink is biocompatible. It will form the biomask and will help
okay
the cells to adhere in that way and grow. It will provide the
okay
ECM also. It will
so if I
generate
create a cavity in between
uh,
and
yeah,
I
basically a
form the
mask.
layers
Yeah, basically a mask is like, I will tell you. It's like you have you seen stickers? Stickers. So stickers, you stick it on and peel it off. Only the portions which are sticky is remaining, right, on your
is
skin.
remaining yes
So it's not a perfect example, but consider the sticky portion as the cavity in which your cells will be growing only. Okay, so the
no
peptide
no
will provide
I am like
location.
if I draw a circle
Yeah.
If
You
I
draw
draw
a
a
circle with
circle,
the
then
peptide ink, then the cells will grow only around the circle. If you do not
around the
fill them,
circle,
yeah, if you do not
in a
fill the circle
circular pattern, they
in a
will grow in
circular,
a
yeah,
circular pattern,
yeah,
and that may form a cylinder. Means if we think about
yeah,
like
yeah, it will form a cylinder if you're seeing in the 3D. 1D, it will form a
3D
circle. But if you're like building
circle.
it up continuously, then it will form a cylinder. But it will be hollow inside
Hmm.
if you do not fill it inside.
Huh.
Okay.
Yes, yes,
Yeah.
yes. Got it. Yeah.
Yeah.
Hmm. Got
Okay.
it. Got it.
Okay. Okay. Thank you. Any
So
other question?
that so vertically we can grow the cells, right?
Vertically, yeah, 3D culture is possible in 3D.
Because organ printing, when we are talking about organ printing, that needs a 3D printing, right?
Yeah, yeah. Organ printing is 3D printing. So basically, 3D
Hmm.
printing means it will print in different layers. Okay, so it will print
Yes.
in different layers and it will lift it up, definitely. Also, you can use some other, usually to do 3D cell culture, also you can do 3D printing. Like you can create a ECM with a 3D matrix using 3D printers. Okay, it's a lot
Yeah, so
more
because
advanced.
tissue engineering, if you are talking about, then tissue engineering also needs 3D printing, right?
Yeah, yeah. 3D printing. It's all work
Hmm.
about your 3D printer. Mostly the most important thing is developing a bio ink, which is not very easy because for your cell growth, you know that it needs ECM. ECM has a lot of factors, so you have to provide all the factors. So your scaffold, the substrate that we were talking about right now.
ECM is extracellular matrix.
Yes, yes, extracellular matrix.
Okay.
So your cells will grow in extracellular matrix. It has all factors, growth factors, and all. If your cells do not form an ECM, then you have to provide all the factors from outside. Okay,
Mm-hmm.
so these are all factors you have to consider. So you can also develop bioink which has those factors. So it all
Yes,
depends
yes.
on you how you want to like make the thing. Basically, you need to have a 3D printer, you have to develop a bio ink, and then you have to think your application. First thing that should come to your mind is application. What is my application and how should I approach it?
Yes.
Okay, so if I want to grow cells, what kind of cells I want to grow, what kind of factors I have to give. So all of these things you have to continuously think like what factors are needed, what are the requirements for the cell, whether it can grow in normal conditions or it needs some special condition. So all of these things will be needed. Then only you'll be able to develop something. So an organ, you can understand how difficult it will be to develop a proper organ, especially like that's why till date vasculated highly vasculated organs have not been 3D printed yet. Till now only skin, some bionic ear you will find, but you will not find only one example I think was given in the module, that was about some plant leaf which was leached and then it was re engineered to form heart tissue.
Heart tissue.
Yeah. There was no example of that. I think there was an example in the module.
My few modules are remaining from
Okay,
week
okay.
3, so I
Okay, you
don't
will
know.
find it in the module. It will be there in the module. So they have taken researchers. What they did was they took a plant leaf, they leached the leaf, and then they reprogrammed the some genes so that it expresses heart tissue. So the leaf started to develop vessels like heart. Okay, it started to
Okay.
beat like a heart. So so that could be one of the ways you can address how you can make highly vasculated organs, because plant, as
Yes.
you know, it has very nice vein system.
Nice. Yes, it is highly
Yeah,
vasculated. Yeah.
yeah, vein system. So that is what they thought maybe they can do to make
Hmm.
uh, vasculated,
Heart
very
tissue.
complicated, yeah,
Very.
very complicated
Hmm.
organs.
Okay. Thank you.
Okay, welcome. Any other questions? Any other questions? Anybody has any other questions? Okay. I think one of the sirs asked me yesterday regarding whether you can give exam this like give fresh exam. You accidentally enrolled for last year's exam or something.
Yes, ma'am. Yes, ma'am.
Yes, yes.
Actually asked this question.
Yes. So my senior, I asked my senior, who is the TA. He told me, told me to tell you to mail this mail ID. I will write the mail ID in the chat box. Kindly note it down, and you can write a mail to this mail ID regarding your issue, and I think they will be able to address it from their side.
Ma'am, if you can please mail the. Achha, just a second.
You
Suppose.
want me to mail you this mail ID?
Yeah, yeah, yeah.
Okay,
Or
fine.
you can. Yes,
Okay,
ma'am.
fine. I will write a mail and I'll mail you this mail ID. No problem.
Okay, ma'am.
Okay, okay. Thank you. Okay, so if nobody else has any other question regarding today's session, then and regarding any course content, then today we will be closing the session. And it was nice interacting with all of you today. And next week, Saturday, we will have the last session. So if you have any doubts before that, you can ask me. I will try to address them as much as possible, and I will come with a few more extra information modules. I'll share this PPT also. Last class PPT I've already shared in the drive. If you got the drive link through the mail, then you will find the week 2 PPT there. Week 3 also I'll upload right now. And hope you're enjoying this course, and thank you for joining today. I hope that you will join next week Saturday at the same time also. So thank you everyone. Have a good day. Bye bye. You can leave.
Thank you.
Thank you.
Thank you, ma'am.
Thank you.